I will be explaining about a paper published by a group of researchers based in China titled VGAS, a viral genome annotation system. Link will be provided in the description that can direct you to the paper. To start out, there are a few prerequisites I recommend viewers have an understanding of before proceeding with this video. Hopefully you have already taken your second course of introductory biology where you have been taught a little about molecular biology. You know the central dogma of how DNA can be replicated and transcribed into RNA, and how it is further translated into proteins. You have been introduced to the concept of viruses, of what it is and isn't, and how they can replicate themselves using a host's molecular machinery. Basic probability. Knowing that the probability of you flipping a coin on a certain side is 1/2, or 0.5 in decimal form. Or you rolling a certain side on a die is 1/6. Or 0.1666 in decimal form. Another prerequisite is also basic algebra, with only the most complex arithmetic operation you need to know how to do is squaring and square rooting. And knowing how to graph. After this video, I hope to expose you to the fields of and teach you a little bit about bioinformatics, virology, and hyperdimensional space. Now let's begin. This paper provides a new way to annotate viral genomes. Annotating genomes means to put a label on all the genes that are within an organism's genome so that we can better understand what protein a certain organism has the ability to produce and to identify new genes that have not been studied or found yet. Generally, the way most genomes are annotated is by taking the entire genome and using the BLASTX algorithm to identify all of the open reading frames, or ORFs, then translating it into protein sequences. And then the protein sequences are searched against the RefSeq database to categorize the ORFs from the translated protein sequences as either annotated genes or unannotated genes. An example being, let's say there is a newly discovered animal we haven't seen before. We would take a sample of its DNA, then sequence its entire genome. We would take that genome and run it through the BLASTX algorithm, which will grab all of the open reading frames. Translate it into protein sequences. Then search in the RefSeq database to see if there are any matches. The RefSeq database is a national database where scientists all over the world contribute by inputting proteins and genes they have found in a shared collection. In real life, the database is much larger than what I can animate in this simple example. There are hundreds of thousands of genes in the actual RefSeq database. If there is a protein match within the database, the ORF which the protein was derived from is categorized as an annotated gene and labeled appropriately as to what protein that gene codes for. If there are no matches, the ORF is considered unannotated and means that nobody has submitted this protein sequence to the database before, therefore not much is known about this gene. That is how most organisms' genomes are annotated. However, viruses are very tricky in a sense that they are not even considered as organisms and more closely similar to a biological vessel for means of transporting genetic materials. There are two main problems we have with annotating viruses' genomes using our usual method. First being, they are very small, meaning that they are naturally selected to have a smaller genome and thus also naturally selected to have smaller genes due to the limited size of their genome. Having a smaller gene makes it harder to differentiate between real genes with false positives. In most organisms that have the real estate of having a larger genome, the smaller ORFs are not genes but instead non-coding ORFs that does nothing and exists there just by chance through generations of mutations. To explain this better, let's say you have a three-sided coin, which doesn't exist in real life, but let's just imagine one for the sake of simplicity. Instead of this coin having heads and tails, the faces of this coin can either be green, which indicates a start codon, red, which indicates a stop codon, or yellow, which indicates any of the other codons that it can possibly be instead of the stop codons or start codon. Each faces has an equal probability of turning up, with 33.33% for each sides, if they are not part of a gene. If the codon are part of a gene, then they would naturally be selected in favor of because those that survive would have the gene to pass off to its next generation, and those that don't have the gene will die off. So let's look at this organism with a long genome that looks like this. It is pretty obvious that this portion of the genome does code for a gene, just because intuitively it doesn't look random. It is totally possible that these coins just by chance flipped in a way where we get this order, but is less likely, especially if we see this same sequence over again through multiple generations in its progeny. However, this portion of the genome is kinda iffy. It can theoretically be a gene, but still looks random enough where it is believable that maybe the coins flipped in this order just by chance. But let's look at a virus that has this set of codons. There's less coin for this one because viruses have smaller genome due to their size. This ORF can be a gene, but it also looks like it can also be flipped in this order by chance, just like the earlier example. But the problem is, viruses are naturally selected to have smaller genes while other organisms are not naturally selected to have small genes because they have the real estate available in their genome to carry larger genes which can code for longer, more complex and beneficial proteins. For organisms with larger genome, we can say, well yeah, if there are 5 consecutive yellow coins in a row, it is most likely a true gene and anything less is probably not a gene. With viral genome, we have to lower that threshold to maybe 2 consecutive yellow coin, which increases the false positive rate leading us to count something as a gene, when really it isn't. Remember that this is a very simplified version, and in reality it is much complicated than this because there are not 3 types of codons, but 64 possible different codons, some of which also are redundant in coding the same amino acids, which affects the probability of our quote-unquote flip. There are also other reasons that can cause natural selection to favor some mutation in the genome more than just merely being able to code a useful protein, like docking sites, enhancer regions, repressing regions, or the GC percentage. The importance of this example and the main takeaway is so that you understand why smaller ORFs have a higher chance of being a false positives compared to longer ORFs in the light of probability. The second reason why viral ORFs have a higher false positive rate is because they have overlapping ORFs, with one ORF being on one reading frame and the other being on another reading frame. With having to work with a small genome, it would be favorably selective to have overlapping genes so more genes can fit in the virus limited size. However, overlapping ORF leads to 2 possibilities, either both of the ORFs are true genes, or one ORF is a true gene and the other being a non-coding ORF. When the overlap of the 2 ORFs are large, it is most likely that one is a gene and the other is a non-coding ORF. When the overlap of the 2 ORFs are small, it is more likely that both of the ORFs are true genes. However, when the overlap is somewhere in the middle, it is less clear which possibility is true and where you should set the threshold on defining when the overlap is too big or too small. Both reasonings causes an overcount of ORFs as true genes and thus a method needs to be used to cut out any false positives. This paper offers an alternative algorithm to use on viral genome to cut down the number of false positives. The pipeline of its method is shown like this. First we grab the viral genome, of course. Then we look at all reading frames, 3 reading frames in the forward direction, and 3 reading frames in the reverse direction, to grab all the ORFs. Then we pick out our seed ORF, and use that to derive 5 new ORFs. The seed ORF will be our longest ORF and will act as the positive control. We pick the longest ORF as our seed ORF, the positive control, because the longest ORF is the most likely to be a true gene. We then derive 5 new ORFs from our seed ORF by reading the seed ORF in the other reading frames. 2 reading frames from the forward direction, and 3 reading frames from the reverse direction. These new 5 ORFs which was derived from our seed ORF will act as our negative control, as these ORFs have the least likelihood being a true gene. Why is this true? Well, 1, because these 5 derived ORFs don't even have proper start and stop codon pairs. And 2, remember how we said overlapping ORFs that have huge overlap are least likely to both be true genes, and it is most likely only one of the overlapping ORF to be a true gene while the other is not a true gene. So, we will use our positive control and our negative controls to compare with the candidate ORFs, which are the ORFs that are still left over, that we are trying to rule out any potential false positives so that we only annotate the true genes. And to compare our ORFs with one another, we need to quantify each ORF. Quantify meaning that we measure them, representing one aspect of their quality, into a numerical value. But how, you might ask, do you quantify something so non-numerical as strings of letters? And what exactly is there to even measure from a string of letters? That brings us to the next step of our pipeline, and what I find most interesting. We quantify each ORF by extracting a total of 45 Z variables from each ORF. The Z variable is what we are quantifying our ORF into in order to distinguish them apart, and what we are measuring is in terms of the frequency of nucleotide appearance based on its codon position. We then compare the quantified candidate ORF with the quantified positive and negative control by taking the distance between each candidate ORF to the controls in order to know which candidate ORF to eliminate as a potential false positive. In order to better explain this, let's run this step using only one ORF first. We'll use the seed ORF, or the positive control, and quantify using these two monstrous formulas. I know these formulas may seem very intimidating at first, especially if you're an undergraduate student, but it's not as bad as it looks, and I think once you understand it conceptually, you'll have a better understanding and appreciation of hyperdimensions and non-Euclidean distances. And I'll break it down in simple parts and guide you as to what the formulas are doing. Let's look at only the first formula first and run our seed ORF through it. Let P be a point in 3D space. And to find P, you will need three coordinates, one in the X direction, the Y direction, and the Z direction. And this line just means that in the X, Y, and Z axis, it is limited between 1 and -1. And this will make more sense later on. The letter I represents the codon positions. If you remember in introductory molecular cell biology, genes are read in codons, chunks of 3 nucleotides at a time. Therefore, let's split our seed ORFs into every 3 nucleotides to visualize the codons better. So when I is equal to 1, we are looking at the first position of each codon. When I is equal to 2, we are looking at the second position of each codon. And when I is equal to 3, we are looking at the third position of each codon. A is equal to the frequency of adenine, and there is a total of 5 adenine in this ORF. G is the frequency of guanine with a total of 9. C is the frequency of cytosine with a total of 6. And T is the frequency of thymine with a total of 7. Now let's run our seed ORF through the formula when I is equal to 1. So when I is equal to 1, we will be solving for P sub 1. And to solve P sub 1, we need to solve X, Y, and Z sub 1 by filling in each expression. Let's start with X sub 1. Our first variable is A sub 1. Remember, sub 1 means that we are only looking at the first codon position, and A is asking for the frequency of adenine. So the variable A sub 1 is asking what is the frequency of adenine in the first codon position. There are 2 adenines in codon position 1, out of the 9 nucleotides in codon position 1. Therefore, the frequency of the variable A sub 1 is 2 out of 9. And saying the frequency is similar to saying the probability of. Given that we only look at the first codon position, the probability of me choosing randomly that I pick an A is 2 out of 9. Therefore, we can either say the frequency of A sub 1 is 2 over 9, or we can also say that the probability of A sub 1 is 2 out of 9. Which I just want to mention because it will make things clearer later on. However, now let's do the same for the other nucleotides to get the other variables. In G, there are 3 guanines in codon position 1 out of 9 nucleotides in codon position 1. In C, there are also 3 cytosines in codon position 1 out of 9 nucleotides that are in codon position 1. And T, there is only 1 thymine out of 9 nucleotides that are in codon position 1. Now we can simplify the expressions for each dimension. Dimension being the X dimension, which will represent the X coordinate, the Y dimension, which will represent the Y coordinate, and the Z dimension, which will represent the Z coordinate. Now we can successfully plot our point P1. X sub 1 is equal to 0.11, so we will move our point on the X sub 1 dimension by that amount. Y sub 1 is equal to 0.11 also, so we will move the point up by 0.11 in the y sub 1 dimension. And z sub 1 is equal to minus 0.33, so we can move that in the z sub 1 dimension by that amount. And now we have successfully plot P1 in a 3D space whose position can be located by our coordinate 0.11 in the x sub 1 dimension, 0.11 in the y sub 1 dimension, and minus 0.33 in the z sub 1 dimension. But remember that this is only when I is equal to 1, we still need to do when I is equal to 2 and 3 as well, and that is only for our first formula. So let's do the same thing we did when I was equal to 1 for when I is equal to 2 and 3 also. Now we have 3 points, P1, P2, and P3, which represent the positive control ORF using the first formula. Notice that P1, P2, and P3 occupy in different three-dimensional spaces. This is because P1 is located in the axis x sub 1, y sub 1, and z sub 1 dimensions, while the P2 is located in the axis x sub 2, y sub 2, and z sub 2 dimension, and likewise with P3. Moving in the x sub 1 dimension versus the x sub 2 dimension are as different as moving vertically and horizontally in real life, but mathematically speaking, we only represent them both as horizontal axes because our brain can only visually comprehend a maximum of 3 total dimensions at the same time, and in this example we have a total of 9 dimensions. But just keep in mind that axes with different names are considered different dimensions, and each dimension are just as different to one another as going left and right is as different as going up and down. Now let's move on to the second formula, which is actually the same as the first formula, except with one extra layer of complexity. Just like the first formula, we have a point, P, which is represented by a total of 3 dimensions. Lowercase p just stands for the probability of, which we can honestly just ignore because it is exactly what we did in the last example. The probability of whatever we are calculating will be inside the terms. Capital X stands for the nucleotide that we will be focusing on when taking the probability of. And K will actually stand for 2 things. The first digit of K will be the codon position that we are calculating the probability for, and the second digit of K will stand for the codon position we are looking in reference to. For example, when K is equal to this, we're not saying that K is equal to 12, but that K is equal to 1 too. We are looking at the first codon position to calculate the probability of whatever capital X is equal to, in reference to the second codon position when the second codon position is equal to whatever these nucleotides happen to be when we are calculating the terms. The first digit of K is meant for capital X, and the second digit of K is meant for the nucleotide in the variable that is adjacent to X. Just like with the first formula, I think the best way to understand this is to run it through with the positive control ORF as an example. Let K equal to 1 too. And let capital X equal to A. In order to graph P sub 1 2 super A, we'll need to solve for each dimension like before. So let's solve the dimension x sub 1 2 super A first. And so, we'll fill in our first variable, which I'll underline here. Now let's look at the second digit of K, which is 2, meaning we are referencing to the codon position 2 of our ORF. And the second digit of K is in term of the nucleotide that is adjacent to capital X in our variable, in this case it is A. So we are only going to reference to the adenine that are only on the second codon position, and this gives us a total of 2 adenines that are on codon position 2 that are present. Now looking at the first digit K, which is 1, we are going to look at the codon position 1 on the codons that the blue arrows are present only. And remember, capital X is equal to A, so we are going to count how many adenines are on codon position 1 if the second codon position has an A in it as well. In this case, there are 0 adenines. So the probability that the first codon position has an A if the second codon position has an A is 0 out of 2. Out of the 2 codons we are referencing to, there are 0 adenines present in the first codon position. Now let's solve for the second variable for the X dimension line. We'll look at the second digit of K, which is 2, referencing to the second codon position. And the nucleotide we are referencing is G this time, because that is the nucleotide that is adjacent to capital X. We get rid of all the blue arrows pointing to the codon position 2 sites unless it points to G. We now go back to the first digit of K, which is 1, referring to the codon position 1, and we are calculating the frequency of the nucleotide of capital X, which is still equal to A. The frequency of A on codon position 1 if the codon position 2 is AG is 0 out of 3. We move on to the third variable of the X dimension component, where the frequency of picking an A on codon position 1 if codon position 2 is a C is 0 out of 2. The fourth variable for the X dimension, the frequency of A being on codon position 1 if codon position 2 is a T is 2 out of 2. Now we can solve for the Y and Z dimension components. But if you noticed, we already solved for all of the variables in the X dimension. And that the variables in the Y and Z dimensions are the same as the variables in the X dimension, but the operations of addition and subtraction are the only differences. We already know that P sub 1 2 AA is equal to 0 over 2. P sub 1 2 AG is 0 over 3. P sub 1 2 AC is equal to 0 over 2. And P sub 1 2 at is 2 over 2. So we can just fill in our variables and simplify the equations. With all of the dimensional components filled out, we can plot the point P sub 1 2 super A in a 3-dimensional space like what we did with the previous formula. However, that was only for when capital X was equal to A. We still need to do when capital X equals to T, G, and C, as well as when K is equal to 2 3 and 3 1. Let's quickly go through the rest when K still is 1 2, but when we change capital X to the other nucleotides. The probability that T is in codon position 1 if A is in codon position 2 is 1 out of 2. The probability that T is in codon position 1 if G is in codon position 2 is 0 out of 3. The probability that T is in codon position 1 if C is in codon position 2 is 0 out of 2. The probability that T is in codon position 1 if another T is in codon position 2 is 0 out of 2. Now we simplify again and plot the point P sub 1-2 super T. The probability that G is in codon position 1 if A is in codon position 2 is 1 out of 2. The probability that G is in codon position 1 if G is in codon position 2 is 1 out of 3. The probability that G is in codon position 1 if C is in codon position 2 is 1 out of 2. The probability that G is in codon position 1 if T is in codon position 2 is 0 out of 2. Simplify and plot point P sub 1 2 super G. probability that C is in codon position 1 if A is in codon position 2 is 0 out of 2. The probability that C is in codon position 1 if G is in codon position 2 is 2 out of 3. The probability that C is in codon position 1 if C is in codon position 2 is 1 out of 2. The probability that C is in codon position 1 if T is in codon position 2 is 0 out of 2. Simplify and plot point P sub 1 2 super C. Now we do the same thing again except when K is equal to 2 3 instead. When capital X is A. The probability that A is in codon position 2 if there is another A in codon position 3 is 0 out of 1. The probability that G is in codon position 2 if A is in codon position 3 is 1 out of 3. The probability that A is in codon position 2 if C is in codon position 3 is 0 out of 1. And the probability that A is in codon position 2 if T is in codon position 3 is 1 out of 4. Simplify the equations and plot the point P sub 2 3 super A. We'll repeat again when X is equal to the other nucleotides like before. And just like before, we'll repeat everything for all of the nucleotides again, but this time when K is equal to 3 1. Now from that one open reading frame, we have created 15 different points plotted in their own three-dimensional space. And what we want to do now is to take the vector sum of all of them, creating a single point in a higher space with 45 dimensions to represent our single-seed ORF. In order to explain what it means to take a vector sum and appreciate higher dimension math, it's best to start out with the most simple example and work our way up. Let's start with one dimension, a simple number line that is in the x dimension. What would be the distance between these 2 points? You'd probably say 7, which is right, but how did you get to that conclusion? Perhaps you just counted the number of units between the 2 points. But how would you express that mathematically? To express it mathematically, you can say that you are taking the difference of x2 with x1, where x2 is the point on -3, and x1 is the point on +4. And you can see that we get the same correct result when we plug them in except for the negative sign. To solve this, let's also square the difference and square root it right after. Just like how addition and subtraction are opposite with one another and cancels each other out, squaring and square rooting are also opposite operators and will cancel each other out as well, giving us the same correct answer. The only difference is that by doing this we will always get a positive number, because distance is always positive. It doesn't make sense for me to say I am -7 feet away from you. So you can see that the formula to find the distance between 2 points is very simple with just a hint of complexity by adding the 2 opposite operators. Now let's add another dimension, and let's add this dimension vertically so its origin with the x dimension is perpendicular. meaning that the two dimensions when intersected forms a 90-degree angle at a single point. And let's call this vertical dimension the y-axis and the horizontal dimension the x-axis. You can see that we have now formed a graph. Let's move the blue point in the y-axis by 4 units positively. And let's move the red point in the y-axis by 5 units in the negative direction. How might you find the distance between the 2 points now? Let's look back at our first general equation to find the distance in one dimension. And let's add the same squared term of the x-coordinate under the square root, only replacing the x-coordinate variables with the y-coordinate variables this time. And you can see a familiar distance equation you might have learned back in middle school. The distance between 2 points in a 2D graph is the difference of the x-coordinates between the 2 points and the difference of the y-coordinates between the 2 points, independently squared, then summed together, and then square rooted. Where the blue point coordinates are represented by x2 and y2, or -3 and +4, respectively. And the red point coordinates are represented by x1 and y1, or +4 and -5, respectively. By plugging the coordinates into our 2D distance equation, we can find the distance between the blue point to the red point, which is approximately 11.40 units. Let's add another dimension, and let's add this dimension in the z dimension this time, giving us a z-axis to work with. And you can see, this new dimension is also perpendicular to the x and y dimensions as well. Let's move the blue point -5 units in the z direction, and the red point +2 units in the z direction. Now the new position of the blue point in this 3D space can be found using the coordinates -3, +4, -5, and the new position of the red point can be found using the coordinates +4, -5, +2. Where the first number of the coordinate represents how many units that point moved in the x direction, the second number of the coordinate represents how many units that point moved in the y direction, and the third number of the coordinate represents how many units that point moved in our new z dimension. To get the distance between our 2 points in 3D space, we take our distance equation from 2D space and add the extra z-dimension component into it, like what we did when transitioning the distance equation from 1D to 2D previously. Just like before, we plug in our coordinates for the blue point into x2, y2, and z2. And we plug in our coordinates for the red point into x1, y1, and z1 in order to get the distance in 3D space, which is approximately 13.38 units. So you recall how to solve the distance between 2 points in one dimension is this equation. And to solve in 2 dimension it is this equation. And to solve in 3 dimension it is this equation. If I were to told you to add another dimension, a 4th dimension, onto our 3D plane, you would be correct to guess that the equation to find the distance between 2 points in this new 4D space would be to add another term under the square root. So the position of a point in 4D space can be found using a 4-number coordinate system, one representing the x dimension, the y dimension, the z dimension, and in the new w dimension. And this pattern of finding the distance in higher dimension extends on to infinitely many dimensions as you add one on top of each other. I also want to note that this 4th dimension is added orthogonally onto the other 3 dimensions. Orthogonally, which is a fancy way of saying 90 degrees or at a right angle, but in a more generalized term. The x, y, and z axis are all orthogonal to one another because they form right angles with each other and meet at the origin only. The same goes with this 4th dimension and any other dimension we add on top of it. It just seems that this 4th dimension is slanted and forming a typical line slope in 3D space because it's hard for our brain to comprehend higher dimension space unless it is 3 dimensions or less. But in mathematics we can use our imagination to just say this 4th dimension is no different than any of the lower 3 dimensions, and apply the generalized pattern we observe in 1, 2, and 3 dimensions into higher dimensions or hyperspace. Hyperspace generally meaning any space that is higher than 3 dimensions. And so back to our example. We have these 2 equations. The top one will quantify our open reading frames into 3 points in their own 3D space, and the bottom equation will quantify our open reading frames into 15 points each in their own 3D space too. Let's represent these 2 equations into one function. Remember, we are still working with the positive control seed ORF, and we use this seed ORF as an input for our function, resulting in multiple outputs for a total of 18 points each in their own 3D space. 3 points from our top equation and 15 points from our bottom equation. Now we are going to take the vector sum of all of our points together. Meaning we are adding all of our 3D space which we obtained and adding them on top of one another orthogonally, creating a hyperspace with 48 dimensions. Each 3D space which our point was represented in had 3 dimensions, and we had a total of 18 points. 3 times 18 equaling to 48 dimensions in total, which we added on top of one another. Now we have this new point which represents our seed ORF in 48-dimension hyperspace. And this point in hyperspace can be found with a 48 number coordinate. We do the same for our 5 negative controls as well, putting them through our function to obtain 5 points in the same hyperspace that our positive control exists in. We then find the average distance of all 5 negative control points to form one negative control point that represents the average. Now we grab our candidate ORFs, the ORFs that we aren't sure if they are true genes or not, and run it through our function one by one to obtain a point in our hyperspace. We then compare the distance between our candidate ORF with our positive control and our negative control. If we find that the distance between our candidate ORF to be closer to our positive control in comparison with our negative control, we put this under as our preliminary ORF. If we find that the distance between our candidate ORF to be closer to our negative control compared to our positive control, we abandon this ORF, giving us our first cutoff to reduce the amount of viral genes that are false positives. This first cutoff filter addresses the problem with our 3-sided coin example earlier. By comparing the candidate ORF with the positive and negative controls in terms of nucleotide frequency based on their codon's positioning, which its frequency was represented by our hyperdimension space, we have a more selective way of picking our ORF that is not based on the gene's length, but rather how similar the candidate ORF is to our positive control than our negative control in terms of this frequency. So, by using the function to quantify our ORFs into a point in hyperspace, or how the paper describes it, into 45z variables, then comparing the candidates with our controls by measuring its Euclidean distance between both controls, we end up with a list of our preliminary predicted genes. We then take our preliminary list and use the BLAST algorithm on it to compare the ORFs with the RefSeq database to see if there are any matches. ORFs can be categorized into 3 different buckets, ones with high homology, low homology, or medium homology. ORFs with high homology with a reference gene in our RefSeq database means that our candidate ORF's translated amino acid chain matched with a known protein's amino acid chain 100% or with high similarities. Since there was a match that surpassed a high similarity threshold, we can safely say that this ORF is most likely a true gene that this virus does code into a protein in real life. ORFs with low homology means that our candidate ORF's translated amino acid chain did not match or were even similar to a known protein's amino acid chain. We then abandon this candidate ORF, saying that it is most likely not a gene that codes for a protein and instead is an artifact of chance, in that multiple mutational events throughout this ORF's generation made it appear to be a gene by having a start and stop codon, but really is not a gene. ORFs with medium homology means that the ORF is similar to a gene on the RefSeq database, and that this similarity could possibly be due to overlapping ORFs, and therefore need to account for that. As mentioned before, there are 2 possibilities when an ORF is overlapping a true gene. Either this blue ORF is a true gene that is also overlapping the another true gene indicated by the red arrow due to natural selection favoring viral genes to be overlapped due to their small genome size real estate. Or the blue ORF is not a true gene and instead just a non-coding protein ORF that just so happens to be on top of the actual red ORF, which is a true gene that does code for a protein. This overlap is the reasoning why we obtain a medium homology score. The blue ORF hijacks some of the red's DNA sequence, which is why it somewhat matches to a gene on the RefSeq database. If the overlap between the red and blue ORF was large, it is safe to assume that they are one gene and not double count them. If the overlap is small, it is most likely two genes. When the overlap is in between, we need a way to decide if we are going to count this scenario as one gene or two genes. This paper has decided to put a threshold value so that if the overlap is a length of greater than 1/5 of the true gene's length, 1/5 equaling to 0.2, we would count that as an overlap large enough to say the two ORFs are actually just one gene. If the overlap's length is smaller than 1/5, then we would say there are actually two different genes present. So depending on how large the overlap is for ORFs with medium homology, we will either count them as true genes or abandon them. And that concludes how this paper has developed a pipeline algorithm method to annotate viral genomes in order to solve the high false positive rates of viral genome annotation, called VGAS.